Welcome to the fifth lecture on the lecture series on the enzymes used in recombinant DNA technology. Today we are going to learn about the alkaline phosphatase, or it is also called as the alkaline phosphomonoesterases. Alkaline phosphatases, it is going to remove the 5' phosphate from DNA and the RNA, that is, it carries out the dephosphorylation reaction. It is also able to remove the phosphate from the 3' end. Now, as it is going to break down the monoester bond, okay, and it is going to remove the phosphate group. That's why it is called as an alkaline phosphomonoesterases. And as for the reaction, it is requiring the alkaline conditions. It is also called as the alkaline phosphatases. Okay, that is, in the alkaline conditions, it is able to do the dephosphorylation. That's why it is an alkaline phosphatases. Now here you can see, okay, in this reaction, okay, you can see that here it is a DNA, okay, or it is an RNA. It is having the 5' phosphate. Okay, at the 5' end there is a presence of the phosphate group. So in the presence of the alkaline phosphatases, okay, there is a removal of the phosphate group, so that here you can find that at the 5' end, now who is there? There is a presence of an OH. Okay, so this alkaline phosphatases, it is going to act on the DNA or the RNA, and it is able to remove the phosphate group. That is, it is going to do the dephosphorylation reaction. Here, when you look at the substrate, it may be a single or the double-stranded DNA, or it may be the RNA, it may be the RNTPs, or it may be the DNTPs. Now, these enzymes, they are called as an metalloenzymes, as they are utilizing the zinc as an cofactor. Now, when this alkaline phosphatases, it is going to act on its substrate, the phosphate which is present at the 5' end, now it is going to be accepted by the alkaline phosphatases, and that it is a serine. Okay, so here the intermediate which is going to be formed during the reaction, it is a phosphorylated serine intermediate. Okay, now going towards the next one, and it is a source of the alkaline phosphatase. The alkaline phosphatases, it can be isolated from the bacteria, that is from the E. coli, and it is called as an bacterial alkaline phosphatases. It may be isolated from the calf intestine, so called as an calf intestinal alkaline phosphatase, or it can be isolated from the shrimp. That's why it is going to be called as an shrimp alkaline phosphatases. Okay, now going towards this chart, okay, this chart, it is going to summarize the number of phosphatases they are used, their structure, their cofactors, and its stability at various conditions. Okay, so the BAP it is what? Bacterial alkaline phosphatases. Okay, this it is a glycoprotein, and it is requiring the zinc as an cofactor. Just now we have seen that the alkaline phosphatases, they are the metalloprotein. That's why here there is a requirement of the metal ions, and the metal ion which is required by the bacterial alkaline phosphatases, it is the zinc. And when you look at the stability, these enzymes, they are highly stable at high temperatures, and they are also stable at a high concentration of the detergents. Okay, next one, here it is an CIP. Okay, CIP, it is a calf intestinal alkaline phosphatases. Okay, it is a dimeric glycoprotein, and it is requiring the two cofactors. One, it is a magnesium ion, and the second one, it is a zinc ions. Now, these CIP, they can be inactivated at 65 degrees Celsius for 30 minutes, or you can keep at 75 degrees Celsius for 10 to 15 minutes. These CIP, they are sensitive for the proteinase K. Okay, so they can be cleared with the help of the CIP. It can be cleared by the proteinase K. And here, the CIP, it can be inactivated by use of the chelating agent, and that chelating agent, it is a EGTN. So here the CIP, it is requiring the cofactor magnesium ions and zinc ions. So these zinc ion and the magnesium ions, they can be chelated with the help of who? With the help of this EGTN. Next here it is a SAP. SAP, it is a shrimp alkaline phosphatases. It is a glycoprotein requiring the zinc as an cofactor, and it is going to be completely inactivated at 65 degrees Celsius for 15 minutes. Okay, now coming towards the applications. Okay, so here It is used for removal of the phosphate residues from the 5' termini of the nucleic acid before radiolabeling. So here in this reaction, okay, you can see that here it is an DNA with a 5' phosphate. Okay, here it is an RNA 5' phosphate. So at this 5' end, okay, radiolabeling it is going to be done in during, okay, radiolabeling it is going to be done during the sequencing, and that labeling it is at the 5' end, that it is a end labeling. So when you are doing the end labeling, first you have to remove this phosphate, and then with the help of the kinases, with the help of the polynucleotide kinases, you have to add the radiolabel phosphate. So here first, to remove this phosphate from the 5' end, to remove this phosphate from the 5' end, there is a use of the alkaline phosphatases. A second application, it is what, to remove the phosphate residue from the 5' termina from the fragment of the DNA to prevent self-ligation. Now, here, this it is a reaction. It is going to explain you what has been told in that application. So, when you have to clone a foreign DNA fragment into a vector molecule, the vector it is going to be cleaved. Okay, so this it is a vector which has been which has been digested with the restriction enzyme. So when you observe here, there is a phosphate at the 5' end, and there is a OH at the 3' end. So here, okay, so here in the normal reaction, when you are going to add this foreign DNA with this vector molecule and do the ligation, what may happen? This foreign DNA it can get ligated with this vector molecule, or there may be the self ligation. That is here, okay. This phosphate it can make the phosphodiester bond with the OH in the presence of the ligase. This phosphate it can make the phosphodiester bond with this OH, that is in the presence of the ligase. And joining of the vector molecule to the vector molecule, it is going to be called as the self-ligation. Okay, so here such a type of a self-ligated molecule it may form. So this, to prevent this, okay, okay, if you are going to remove this phosphate group, okay, if you are going to remove this phosphate group, the self-ligation now it is going to be prevented. Okay, now here you are going to treat your vector, okay, digested vector with the help of the alkaline phosphatases, and here what you find that at the 5' end and at the 3' end, on both of the ends, who is there? There is a presence of the OH. So in this condition, okay, in this condition, ligases, it cannot do the ligation. Okay. Now, here you are going to add the foreign DNA molecule. So, in the foreign DNA molecule, now here it is, here there is a presence of the phosphate at the 5' end. Okay, here it is a presence of the phosphate at the 5' end, and here it is what? Here it is an OH. Now, such a DNA fragment, when you are going to add it with a vector, and you are allowing it to get ligated, okay, at that time what you find that this phosphate, okay, this phosphate, it can form the phosphodiester bond with the 3' end. So one of the end of this, okay, one of the end of this foreign DNA, it is ligated with whom? It is ligated with the vector molecule. Then you may go for the transformation, or you may use the polynucleotide kinases and allow the next reaction. Okay, so when there is a transformation, these nicks now they are going to be sealed in the bacterial cell. Okay, as it is going to be found out that there is what? There is a defect into the DNA molecule. So in this way, alkaline phosphatases, they are used to prevent the self ligation. Second, next it is what? It can be used as a reporter enzyme in non-radioactive assays to detect and localize the nucleic acid and the proteins. So here, when it is a southern blot, northern blot, and a western blot, at that time, okay, at that time the labeling, it is a non-radioactive labeling of the probes. And when there is a non-radioactive labeling of the probes, at that time there is a use of the alkaline phosphatases. As alkaline phosphatases, they are the enzymes. They can act on the chromogenic product, chromogenic substrate, and by acting on the chromogenic substrate, it can produce the product which is the colored one. And by observing the color of the product, you can find out that find out that if a foreign DNA it is present, or a foreign RNA molecule is present, or a protein it is going to be present in a given sample. Okay, in this way, there are the applications of the alkaline phosphatases. I think you may have clear the idea about the alkaline phosphatases. If you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.